Hello everybody, my name is Elliot Clark and I've been working in the Spatial Cognition Lab under the direct guidance of Candace Peacock and Mike Starrett. These past 6 weeks I've been engaged in research on the emergent nature of alignment effects in spatial learning and memory. So what is alignment? Alignment is a key component of spatial memory which is used to navigate cities and in everyday cognitive tasks. So an easy way we can assess this is through a JRD, or judgment of relative direction. In a JRD, participants imagine themselves standing in a certain location, facing another location, and then they are directed to point towards a third location. So we can have 2 types of JRDs. We can have aligned JRDs, one of which is shown in blue. So in an aligned JRD, participants are orthogonal to the salient geometry of the environment. So an easy way to think about it is, are they looking down a road? If they are, then it's aligned. We can also have misaligned JRDs in which they are skewed to the geometry of the environment, shown in this orange JRD. So an example showed here on the map of downtown Davis would be this blue JRD would ask participants to imagine themselves standing at Burgers and Brew, facing Panera Bread, and then point towards the Veers Irish Pub. So, what are alignment effects? Alignment effects are a bias to perform better on memory tasks that are aligned with the environment. In my research, we are mainly going to be focusing on JRD, so that would mean that when a participant is undertaking an aligned JRD, they are more likely to perform better on it than if they are undertaking a misaligned JRD. Another key point of information to note is that map learning increases accuracy on JRD tasks significantly more than route learning. Map learning is just studying a map of the environment, while route learning is actually going through it and navigating it yourself. So while much research has been conducted already on what alignment effects are, not much has been conducted on the emergence of them, so we don't really know when they appear. So we are hypothesizing that alignment effects will not be present initially when participants are first navigating an area, but that they will emerge after multiple learning stages of exploring an environment. So we had 2 experiments with 24 participants each. The first experiment ended up losing 3. So as you can see, we had an Oculus DK2 virtual reality headset shown right here. This allows for an immersive virtual reality experience. You have a full range of view. We also have the omnidirectional side width virtualizer treadmill. This has motion sensors down here that will track your movements. Participants wear frictionless shoes so they can walk easily on it. And then we designed our environment in the Unity 3D game engine. So the procedures of experiment 1 and experiment 2 are almost identical. The only difference is that experiment 2 adds a map learning element prior to navigation. So this video shows the navigation stage of the experiment. So a participant will be navigating through the environment. They have free ambulation, so they can walk wherever they want, and they will be directed to find one of 8 stores. So Mike, one of my grad students, is actually approaching one of these stores. And so once he finds the store, he will be directed to find another store. And that will continue until he has found all 8 stores in the environment. After that, he'll be transported to a JRD screen, where he will take a set of JRDs, and then he will repeat this 6 times. So, oh, sorry. Okay, yeah. So after he has found all 8 stores in the environment, he will be transported to a screen like this. So this just has the directions for a JRD. It says imagine standing in the bookstore, facing the camera store, point to the gym. That's shown right here. This is the bookstore, camera store, and gym. This is a map of the environment. And they will take 28 JRDs, 14 of which are aligned with the environment, like the blue one, and 14 of which are misaligned. And they will repeat this and the navigation step 6 times with the same map. So for experiment 2, the map learning element that is added is this right here. So participants will study this map to criteria. So until they are able to complete this click and drag click and drag test of their map knowledge, they will not be allowed to continue to the navigation stage. Unlike the navigation stage and the JRD task, participants only take this map task one time at the very beginning of the experiment. So what we found was that 16 out of 21 participants showed alignment effects in experiment 1, while 13 out of 24 showed alignment effects in experiment 2, and that the average emergence of these alignment effects for experiment 1 was about halfway through block 3, and for experiment 2 it was closer to block 4. So these are far away from 1, so it does not appear that alignment effects are intrinsic. So one thing to note is that individual differences play a key part in this. So this just goes to show the variability of participants in the emergence of alignment effects. So here with subject 325, we see actually there's no emergence of alignment effects throughout the entire experiment, while in subject 322, there is alignment effects do not emerge until very late on block 6, so that's their 6th repetition of exploring the environment. And then in subject 318 here, we see a very early response to alignment effects, so they actually start around block 3 and even block 2 there, you see that disparity. So for analysis, we ran paired one-tailed t-tests for each participant on their misaligned and aligned JRD trials to see if there was a significant difference between the two, and that's how we could tell when alignment effects emerged for them. We only wanted to find the first block in which they showed alignment effects because all the blocks after the first block would just be showing the persistence of alignment effects. Then we obtained the average first block where participants showed alignment effects, and we ran one sample two-tailed t-tests against one to see if the mean alignment effect is different from one. So this will tell us if the alignment effect is actually significant on the blocks that we're checking. And then finally, we'll find the 95% confidence interval where participants show a significant alignment effect, and that's the really important part. So here in experiment 1, let's take a look at the lines first. So the blue lines mean that they are aligned trials, and the red lines mean they're misaligned. So this is a graph of pointing error and testing blocks. So the higher up the data points, the more error there is. So as you can see, there is a curve. So as participants explore the environment more, they get better and better at the JRD tasks. This purple shaded region is the 95% confidence interval in which we are 95% sure that participants will show their first alignment effects. So as you can see, between experiment 1 and experiment 2, there was not a large difference between the locations of the confidence intervals. However, there was a significant difference in their initial performance on block 1. This is because, as stated in the introduction, map learning helps JRD performance significantly more than route learning. So we can see that right there. And so in summary, alignment effects have an emergent nature. We determined that. And prior map learning does improve JRD performance, as shown right here, but it does not elicit an initial alignment effect. So if it did elicit an initial alignment effect, we'd expect this confidence interval to shift to the left and be more around blocks 1 and 2, because that would show that participants are having an earlier emergence of alignment effects. But instead they stay there, so map learning does not affect the emergence of alignment effects. And this actually goes against previous research that has been done in the field. So Tim McNamara conducted an experiment in which participants navigated through an environment multiple times and then were tested on spatial performance, and they found that alignment effects emerged in the first time they were tested. So unlike Tim McNamara, we are actually testing participants after each time that they navigate the environment. And the purpose of this research will help further progress our understanding of spatial representations and how they're formed in respect to alignment. So looking forward, it would be helpful if we could redo this experiment, but with a larger sample size as our current one. As you can see in experiment 2, we only had, it was like 11 participants. And so if we could get 24 participants who all showed alignment effects, we could have a better data set. Another experiment that we could conduct would be a map learning study. So that would be if you took experiment 2's procedure, so map learning, navigation, then JRDs, and you just took out the navigation part. So it was just map learning straight to JRDs. And then we could see if the navigation was affecting the confidence intervals. And then finally, we can check virtual reality if there are significant differences between exploration in virtual reality and the real world. So we can see if there is a difference in the emergence of alignment effects in virtual reality in the real world. And we need an experiment that conducted parallel experiments, one in virtual reality and one in the real world to test that. I would like to thank Candace Peacock, Michael Starrett, who isn't here, and the rest of the UC Davis Spatial Cognition Lab, as well as everybody in the YSP program.
Did they have to have certain requirements?
Okay, yeah. So all participants were compensated with money when they participated in the experiment or course credit. Participants had to be under a weight limit because the treadmill, this treadmill can only support 200 pounds. So if a participant was over 200 pounds, they could not participate. Participants also had to be able to navigate the virtual environment without getting sick. Simulator sickness is a phenomenon that occurs sometimes in virtual reality. So those are some criteria that participants need to meet. There.
This is kind of like going on with what Susan said. Was there an age limit to?
Participants have to be over 18.
Okay.
What are the benefits to testing and or doing these experiments in virtual reality and not the real world?
So in virtual reality, you can control like all the variables. So if you conducted the same experiment in the real world, there would be birds flying around, you'd have people walking around. There are a lot of things that can cloud the data and have there's a lot of outside variables that you cannot accurately take into account. So there'd be a lot more discrepancies in the data. Raj?
Were all the procedures done in one day for like one person?
I'm not sure. Yeah, sorry.
Did the virtual reality did they have like street signs to coordinate with?
No. They had a city environment. It had filler buildings, so there weren't just blank spaces in between the shops. The map right here. So that's all it had. Chris?
In the video of the guy in virtual reality, like he was walking pretty fast. Is that because the video was sped up, or because the actual?
That's because he started running.
I mean, like at the start. Like that, it seems faster than like someone who's walking.
He's just walking fast. Our next presenter will be Eleanor.
Good afternoon, everyone. My name is Eleanor Go, and for the past 6 weeks, I've been working here at UC Davis under graduate student Jessica Huang in the Housing Lab under Dr. Mark Housing, which is in the Department of Neurobiology, Physiology, and Behavior. Okay, so just for some background, in the pancreas, there are tissues called islets, and in the islets, there are 3 different kinds of cells. They are the alpha cells, the beta cells, and the delta cells. And all 3 work together to regulate blood glucose levels. So the beta cell secretes a hormone called insulin, which lowers blood sugar. And then the alpha cell does the opposite. So the alpha cell secretes a hormone called glucagon, which increases blood sugar. And the delta cell comes into play by regulating the actions of both the alpha and the beta cells. The delta cells produce a hormone called somatostatin, which represses alpha cells from secreting glucagon and also represses the beta cells from secreting insulin. And a along with the secretion of insulin, a protein called urocortin-3 is secreted by the beta cell. And essentially what urocortin-3 does is it stimulates the delta cell and causes it to secrete somatostatin. So in this way, the beta cell and the delta cell work together through negative feedback to maintain insulin levels. In type 2 diabetes, there is a loss of urocortin-3 expression. And what this will mean is now that there is no urocortin-3, the delta cell will not have a stimulator, so somatostatin expression will go down. And because somatostatin represses the beta cell, now the beta cell does not have much repression anymore, so insulin will go up. So you might be wondering, if the insulin secretion is going up, why would this lead to diabetes? This is because as the cell is constantly secreting insulin, the cell's machinery gets tired, and then this leads to dedifferentiation. So what dedifferentiation is it's when the cell is unable to express the genes that are necessary for its identity. So what we wanted to do was we wanted to see whether somatostatin would reverse the effects of high glucose on mRNA expression of genes involved in beta cell function. And we hypothesized that yes, it would. So here is a schematic of a beta cell. I'm going to start over here. So glucose goes into the cell from the bloodstream through GLUT2, which is one of the genes that we looked at. And then once glucose goes into the cell, it is metabolized. And during the metabolism of glucose, ATP is produced by the cell. We looked at G6PC2, which is one of the proteins that reverses the metabolism of glucose. So as glucose is being metabolized, it gets converted into this form called glucose 6-phosphate. And what G6PC2 does is it takes glucose 6-phosphate and it turns it back into glucose. So essentially what G6PC2 does overall in the cell is it ups the cell's glucose sensitivity. And once the cell gets ATP from metabolizing glucose, it takes the ATP and it closes the potassium channel. And then once the potassium channel closes, the cell depolarizes. So because it depolarizes, the calcium channels open, and when calcium goes into the cell, insulin is secreted. Now, one thing to notice here is that the opening of the calcium channel leads to insulin secretion and not expression, but we did look at the INS2 gene, which is the gene for insulin. So what did we do? We took a few wild-type mice, and we injected them with enzymes that would break down their pancreas, and that was called collagenase. And then after we take out their islets, we take their beta cells and we cultured them in 4 different conditions. 5.5 millimolar, which is low, 16.8 millimolar, which is high, and then those 2 conditions also with somatostatin. And that was cultured at 37 degrees Celsius for 24 hours. And then after 24 hours, we took them out and we extracted their RNA using a chemical called trizol, and then we performed DNA synthesis using a cDNA kit. And then after we got our double-stranded DNA, we took it to the qPCR machine, which I'll explain next. So what qPCR aims to do is it aims to look at how much DNA you have. So what we did was we took a fluorescent dye called Cybergreen, and we put it together with our DNA into the machine. And when it's inside, it starts replicating exponentially until it runs out of material. But when it runs out, it's not very significant because before that, we established a threshold line, which you can see here, which is a certain amount of fluorescence that represents the amount of DNA. So once the sample reaches the threshold line, a CQ value is established, which stands for the cycle at which the DNA reached the threshold. So essentially what this means is a lower CQ value means that you had more DNA to start with because the DNA reached the threshold quicker. And then the same is true the opposite way around. So a high cycle number or CQ value means that you had less DNA to start with. So in our first experiment, we just decided to look at glucose without somatostatin. So that's just 5.5, which is low, and 16.8 millimolar, which is high. And what we see here is what we expected. So in the low condition, you see that insulin 2, G6PC2, and GLUT2 do not have much of a response. but this is because they aren't supposed to, because 5.5 is low and the beta cell should not be responding. And HPRT is a housekeeping gene, which means that it stays relatively constant and we can normalize to that. And in 16.8, we see that the beta cells, they're compensating, which means that they're upregulating the expression of insulin 2, G6PC2, and GLUT2 in response to the high glucose level. So this is our second experiment, where we added in somatostatin along with the same glucose concentrations. So what we see here is that in 5.5, no matter whether there was somatostatin or not, both cases did not respond as much, so we did not see an increase in expression. And in 16.8, we see that insulin 2 and GLUT2 do not respond, which is kind of strange because in our first experiment, as you saw earlier, they did increase the expression of that gene, but it's kind of inconsistent, and we see that that's not true anymore, except for in G6PC2, where we see that it's consistent with what we had before. So the main thing to notice here is the last condition, which is 16.8 and somatostatin. And with the combination of high glucose and somatostatin, we see that all three genes are upregulated. So what could this mean? This could mean two things. One, somatostatin may increase the expression of mature beta cell markers under high glucose conditions. And two, somatostatin may prevent exhaustion and amplify acute insulin response. However, we cannot make those conclusions concrete because we did see inconsistency between our first and our second experiments, as I said earlier, and we think that this could be because we used different cell plates for growing, and one of them stuck to the bottom, which would cause transcriptional changes. So what we would do in the future is definitely repeat this experiment for consistency. And also, we only looked at mRNA expression, so what this means is that it doesn't really look at protein levels, because beta cells are known to have a storage of insulin, but not actually transcribe and translate it when they sense the glucose. So they release their stores and then they replenish it. So a more accurate way of looking at the beta cell would be looking at protein levels, and this could be done through confocal imaging, which I'll show next. So this is beta cell an islet from an obese mouse in high glucose, and the beta cell is in red, and the green is the calcium. So each time the green flashes, you know that calcium is going into the cell, and this tells you that the insulin protein is being secreted. So this is a proxy way of looking at protein levels. So why would we be doing this anyway? Because in type 2 diabetes, we see that the beta cells are unable to secrete insulin. So if somatostatin can restore the maturity in dedifferentiated beta cells, we could potentially cure diabetes at the root. So without trying to cure the symptoms or anything, we can help the beta cells restore their function. And for type 1 diabetics, this could help them during the honeymoon period, which is in the beginning of the development of the disease, when the beta cells are not all completely dead yet. The body is still able to regulate the blood sugar levels with the help of insulin therapy. So a lot of research is currently being done to try to prolong or freeze this period, and we think that enforcing beta cell rest using somatostatin can help this cause. So I have a few people I want to thank. I want to thank my graduate student, Jessica Huang, for being so patient with me and helping me through everything. Dr. Mark Housing, who is not here, for giving me this opportunity, and the rest of the members of the Housing Lab for being so welcoming. And I would like to thank Corinne Rushing, my counselor, for looking over my papers and answering any questions that I had, and also everyone in the Young Scholars Program.
If somatostatin regulates both alpha 8 and beta cells, how can you just focus on like higher insulin levels and also higher glucose?
Our lab doesn't really look at alpha cells, but I just included that in the Yeah, let's help the triangle. Yeah.
Why do you use mice as a test subject?
Because mice have a really similar body system to humans, and you can easily compare it. And they're easy to obtain, and they reproduce fairly quickly, so we can get a lot of stuff.
What would be a future experiment that you did?
I think one future one would definitely. So we tried to induce beta cell rest using somatostatin, but we can also try inducing beta cell rest using just low glucose and compare the two. So we could put it in high glucose and then low glucose and see how expression changes, and then put it in high glucose and somatostatin and see how that changes, and then compare the two.
Eleanor, over here.
Yeah.
Another question. So how did you isolate specifically the beta cells from the islet cells total that you got out of the mouse?
We didn't exactly isolate the beta cells themselves, but
Okay.
we do take out the islets, and then whatever procedure we do, we know that it's affecting the beta cells because whatever we treat it with is specific to the beta cell. The next presenter will be Victoria.
Thank you.
Yes, I ordered. It's
We're still missing one more. Yeah. We're almost ready. Right? No, we're missing one. I got my question ready. What? I can't. I answered. Thank you.
Good afternoon, everyone. My name is Victoria Yang, and for the past 6 weeks, I've been working in the Castile Lab in the Department of Plant Pathology, researching how organic soils influence tomato resistance to insect attacks. We looked at the Solanum lycopersicum, and this is more commonly known as the process tomato, and it's a major crop for California. However, it is susceptible to many insect vectored viruses, such as the beet mealypot virus. Some symptoms that you may be able to see are that it has curled leaves and the underside has swollen and purple veins. Younger plants infected with BCTV will often turn yellow and die. And while older plants, they won't die, they will be ripening prematurely and their fruit will be too small that's no longer suitable for consumption. BCTV is vectored by the beet leafhopper, as shown here. And current efforts to control it aren't working because the beet leafhopper, after just 15 minutes of landing on the tomato plant, can infect it with BCTV. So farmers are employing chemical insecticides like neonicotinoids, and these aren't effective enough at resisting the insects from coming in the first place. So for my project, I was studying plant-insect interactions, in particular how organic farming affects the interactions between the tomato plant and the leafhopper. Our long-term goal is to apply this knowledge to create a more sustainable insect control that does not rely on chemicals, but still won't compromise crop yield. We had preliminary research from our lab in previous years, and what we saw were that in conventional plots, there are significantly more insects than we were able to find in organic plots. So this led to the idea that organic practices decrease pest populations. In order to investigate if this is the case, we are taking a bottom-up approach to studying this as opposed to a top-down approach. With a top-down approach, we would be looking at biological controls, such as spiders, that would be present on the tomato plants. And these may result in deterring insects like the leafhoppers from coming to the plants. But instead, with our bottom-up approach, we're looking at the health of the soil and other environmental factors that may create stronger, healthier plants that are able to defend themselves and resist insect attacks. For my project, we are comparing the organic and conventional soils. With organic soils, you see they're derived from organic matter, and they have much higher amounts of nutrients and minerals. With conventional soils, though, we see that they're essentially sterile. They don't have a lot of minerals or nutrients, so it is necessary to pump in additional synthetic chemicals in the forms of fertilizers, insecticides, and pesticides. And most farmers are employing conventional farming techniques because it is cheaper and easier to manipulate than the organics. So the question we're trying to answer is, are Solanum lycopersicum, which are the tomato plants, grown in organic soils more resistant to insect attack than the ones grown in conventional soils? For my experiment, I took samples from Russell Ranch. Russell Ranch is a controlled farm on the UC Davis campus land. And if you can see in the blue plots, those are my organic samples. And in the purple plots, those are where my conventional samples came from. Then we conducted 3 different tests, the first being a sweep test. So when we went out into the field, we did 10 consecutive sweeps using a sweep net, and for every 10, we counted that as 1 sample. For each plot, we took 6 samples, and then we take them back to the lab and classify and count the number of insects found in each sweep. What we found was that organic practices do decrease pest populations. If you see in the blue, that is my conventional, that's from my conventional sample, and we saw significantly more insects in the conventional sample than in the organic sample. Next, we had a choice bioassay, and what we did with this is we constructed a capsule. On one end, we had an organic tomato leaf. On the other end, we had a conventional tomato leaf. Then we inserted 5 leafhoppers. These leafhoppers were starved for 2 hours prior to the experiment in order to maximize the chances of them ultimately making a choice and deciding to go after one leaf or another. And then after 2 hours and 24 hours, we counted the number of leafhoppers present on each of the 2 leaves. What we saw were that the conventional leaves are more attractive to the leafhoppers than the organic leaves. As you can see from the graphs, there were significantly more leafhoppers that went to the conventional leaf instead of the organic leaf. And another thing that you may notice is that after 2 hours and 24 hours, the data is very, very similar. What this implies is that after the insects made their initial choice at 2 hours, they, for the most part, stuck with that decision for the remaining 24 hours. And this implies that there was an initial attraction to the conventional leaf that caused them to go there in the first place. So now we're trying to look inside the leaf and see why this attraction occurs. When a leafhopper begins feeding on the plant, it causes the defense system to be activated. So we have, after the defense is activated, we have hormonal regulation, and this activates certain defense pathways that are conserved throughout all plants. For our purposes, we were just looking at the hormonal regulation of 2 very important phytohormones, jasmonic acid and salicylic acid. Jasmonic acid is known to be associated with the physical wounding of the leaf, such as when a leafhopper would probe beneath the cell surface using its stylet and activate the jasmonic acid, and salicylic acid is more known to be associated with pleiotropic pathogenic attack. However, there is significant amounts of crosstalk and interaction between the 2. For our method, what we did was we had Solanum lycopersicum sown in controlled greenhouse conditions. We grew them for 3 weeks and then transplanted them into the field. And we believe that after 3 weeks, that is when they are most vulnerable to insect and pathogen attack. Then throughout the field, we had 12 sites, and at each of the 12 sites, we chose 2 adjacent plants. 1 plant served as our independent variable. So what we did was we put a bag around the plant and then inserted 5 leafhoppers. We did this in order to hopefully elicit a response with the defense hormones. and the other served as a control, so we did not put in any insects. Then we took hormone extractions from the youngest leaf. We chose to do the youngest leaf because older leaves may be susceptible to other biological and abiotic stressors. And then we subjected the hormone extractions to LCMS, which is liquid chromatography mass spectrometry, and we use this to determine the concentrations of desmonic acid and salicylic acid. What LCMS is, it's a combination of liquid chromatography and mass spectrometry. With liquid chromatography, we are able to separate the different components in solution, and with mass spectrometry, we are able to identify the individual components based on their unique masses. So with the combination of both, we are able to determine the concentrations of desmonic acid and salicylic acid. What we saw was quite interesting. So if you would first take a look at the blue graphs, we saw that after we added the leaf hoppers, there was an increase in the concentrations of the desmonic acid defense hormone. And we expected to see this, because when you add the leaf hoppers, it should elicit this defense hormonal response and increase the concentrations. What we didn't expect to see, though, was that it was the exact opposite for the organic plants. The control had a higher desmonic acid concentration than the ones that were added to leaf hoppers. And this was something interesting. And we also saw this trend with salicylic acid concentrations as well, where the conventional follows the trend we expected to see, but the organic is the exact opposite. This is likely because we don't have all of our data yet, so we're assuming that once we get all of our data, we will see a more normalized graph, or at least one with smaller amounts of variation. So while we can't make any conclusive conclusions on the trend, what we can say conclusively is that organic plants overall have higher amounts of the defense hormones desmonic acid and salicylic acid, consistently more than the conventional crops. In conclusion, what we saw was that organic practices decreased pest populations. We saw this from the sweep test, and then from our choice bioassay, what we saw was that the conventional leaves are more attractive to the leaf hoppers, or that the organic leaves deter the leaf hoppers more than the conventional leaves. And we also saw from the hormone extractions that organic practices increase defense hormone levels over the conventional practices. We hope that in the future we are able to develop the next generation of insect control, shifting from chemical insecticides and pesticides to an insect control that is taking advantage of natural plant defense systems and hopefully won't rely on the use of chemicals as much, which will lead to a healthier future for both humans and plants. I'd like to thank the Castile Lab, in particular my PI, Dr. Terry Castile, and my mentor, Rob Lundell, my counselor, Sarah, for all of her help and encouragement, and the Young Scholars Program for the amazing memories and this great experience. So do we have any questions?
For the organic plant, why do you think that the ones without the leaf hoppers actually had more defense hormones?
So are you asking why with the hormone?
Why you think so? So your hypothesis.
Oh, why there would be more in the control? Well, we can't conclusively say that that is the truth because we don't have all of our data. But if it does end up being the case, we would want to definitely go back to the fields and repeat this experiment. It may be because of stressors in the plants that cause this difference, or there may also be some errors in the practice where maybe there did end up being some insects getting into the control plants. Oh, yes.
So how dangerous is BCTV?
So BCTV is extremely destructive to the crop, to the tomato crop, as well as the other crops. In 2013, that was one of the most destructive years in which 1 million tons of the tomato crop were lost in just California due to BCTV. Camille?
For the bioassay, how are you sure that it was just like the leaf that caused the X2O instead of something else?
Okay, so we did put controls on the capsule that we made. So one thing that we did was we alternated between the right and the left. So we tried to make sure that direction wasn't one of the causes of why it would go one way or another. And we also covered it with a black tarp for the entire duration of the experiment to make sure that they weren't attracted to one side because of light.
So if SA was upregulated with pathogens, why did you see an increase?
So that's actually a really good question because we expected that with the leafhoppers causing damage onto the leaf, that there would be more of a jasmonic acid increase and maybe a constant with the salicylic acid. One reason that both were increased was because there is significant amounts of interaction and crosstalk between both of the phytohormones, and also because with the physical wounding on the leaf, that may allow other pathogens to enter the leaf, and that may also be a reason as to why the salicylic acid concentrations increase.
Okay, we're good.
Next up, we have Raj Ajudia.
Hi everyone, my name is Raj Ajudia. I'm from Folsom, California, and this summer in the Pinkerton lab, I worked on tracking the dynamics of mesoporous silica nanoparticles in the lungs using fluorescent immunohistochemistry and confocal microscopy. Nanoparticles, which are particles that have at least one dimension less than 100 nanometers in length, have already been used in a wide variety of consumer and industrial applications, including cosmetic, food, automotive, and other products. However, scientific attention is now being turned to its possible application as a material for a drug delivery system that can accurately and efficiently send drugs to deeper than ever target sites in the lung. Of the different types of nanoparticles being studied for this purpose, such as carbon nanotubes, polymeric micelles, and quantum dots, particular attention is being given to the mesoporous silica nanoparticle, or MSN. MSNs have several properties that make them such suiting vehicles for drug delivery, including their favorable chemical properties, their thermal stability, and biocompatibility. Additionally, the very structural and textural properties of MSNs including their large pore volume, high surface area, and narrow pore size distribution, make them so easy to pack in medicine and subsequently release the medicine at the very target sites in the lung. So exposure to MSNs in both humans and mice takes place in a very similar process. First, the MSNs go to either the oral or nasal cavities. From there, they travel down the trachea, through the bronchi and the smaller bronchioles in the lung parenchyma, and all the way down to the alveoli. This is an alveolus. The alveoli are the basic units of respiration in the lungs, and they're most significant for their role in gas exchange. There are 4 types of alveolar cells: type 1 epithelial cells, type 2 epithelial cells, and 2 types of phagocytic cells: dendritic cells and alveolar macrophages. Each of these alveolar cells has a different function in the lungs, but of significant interest to our research are the alveolar macrophages and the dendritic cells, which have their roles as the resident soldiers of the immune system. In rats and in mice and humans alike, the immune system has a responsibility of protecting its host from a variety of environmental agents, such as microbes, chemicals, and particles. The initial actions taken by the immune system to battle these environmental agents can be comprised into 2 steps, 2 primary phases. First, the recognition of self and non-self. Basically, what belongs in the lungs and what doesn't. And second, the consequent response. What does the immune cell do if it recognizes something that doesn't belong in the body? The immune response itself can also be classified into 2 sections: innate immunity and adaptive immunity. Innate immunity is immediate, but it's basic response to the foreign bodies. Alveolar macrophages are an example of an innate immune cell. On the other hand, the adaptive immunity represents the immunity that takes time to build, but when it is built, it's a highly specific, highly targeted, highly prepared attack to the foreign body. Dendritic cells are an example of an adaptive immune cell. So, knowing that MSNs are ultrafine particles that can go really deep into the lungs, our primary concern is that can they go so deep that they inflict inflammatory damage in the lungs, which could potentially be very, very dangerous? Well, fortunately, the answer is no. Previous research in the Pinkerton lab has shown that MSNs do not trigger inflammatory responses, which is good because now the second question in MSN research is knowing which cells in the lungs take up the MSNs. And moreover, over a period of time following exposure, do the MSNs go to different cells in the lungs? But I would be remiss if I said that we had absolutely no clue about this. Previous studies have shown, like in the image on the left, previous studies have shown that MSNs do in fact conglomerate around these blue dappy-stained bodies, which are alveolar macrophages. Thus showing us that alveolar macrophages do take up the MSN. However, these images are taken of bronchoalveolar lavage samples, or BALF. The way these work is that at the time of necropsy for the mice, liquid is pushed down into the lungs, where cells and MSNs and all that conglomerate in the liquid, and the liquid is pulled back out. This is the images on the left are taken of those samples. So, although they show us that MSNs do in fact go around the alveolar macrophages, they don't preserve the very long tissue structure that's needed to know about the dynamics of the MSNs. Where do the MSNs travel over a period of time? And that's why our research is unique, because we're able to preserve the actual long tissue structure of the samples and know where the MSNs travel exactly in the lungs over a period of time. So how do we do this? The first step in all this is to expose the lab mice, to MSN. We did this for 5 hours. And after that, after 1, 7, and 21 days post-exposure, the mice were necropsied and their tissues were preserved and sectioned onto slides for fluorescent immunohistochemistry. So the way fluorescent IHC works is that it's based off the fact that each cell of interest in the lungs has its specific protein. For example, for epithelial cells, that's CDH1. For alveolar macrophages, it's CD11C. And for dendritic cells, it's cyclic F. These proteins, because we know each of these cells has this specific protein, we can find primary antibodies that will attach to the antigens on these proteins. So that's the first half of IHC. We get the primary antibodies and we apply them to the antigens on the specific proteins we're looking for. The second step in IHC is then creating a secondary antibody that applies to the primary antibody. But there's a catch to this antibody, and it's that a fluorophore is attached to it. A fluorophore, if you guys remember from lecture, is something that when UV light is shined upon it, it fluoresces a certain wavelength. So the fluorophore that we use, or the secondary antibody that we use for our research, streptoavidin 408, it emits a certain fluorescence that our confocal microscope can see. And so how the confocal microscope works is that it can focus on a specific wavelength, which is for us the wavelength that SA408 fluoresces at, and basically take images of that. The actual handling of the microscope was pretty simple. First, the medium of refraction had to be put on the slide, usually oil for us, and then it was locked into the microscope, and then we were able to adjust the lens to figure out, to see the images and kind of clear that up. Yeah, it's really simple. I didn't try getting too close to the microscope because it's close to the cost of a brand new Lamborghini. So I opted to stay closer to the monitor, which is cool on its own because the way the images come out of the microscope, the way the microscope takes the images of the samples, is that similar to how magnetic resonance imaging takes place in MRI scans, how that works is that in an MRI scan, different sections of the body part being scanned are taking different pictures at the different slices are taken. And all those pictures together make a 3D image. So similar to that, the way the confocal microscope works is that it takes pictures at different slices of the tissue on the slides and makes that into a 3D image. We call that Z-stacking. And you'll be able to see a really cool Z-stacking diagram in Chris's presentation. But with that in mind, let's get to the results. So we have images from day 1 samples, day 7 samples, and day 21 samples. But they all show the same thing, which is that, actually before I get into that, let me explain what the colors mean. Blue represents the DAPI stain. So DAPI stain stains onto the cell body or the cell nuclei, and for us that was the alveolar macrophage. Those were the nuclei being stained. So for us, blue equals alveolar macrophage. Green represents the protein CEH1 that the secondary fluorophore is attached to, and that's for the epithelial cells. So green equals epithelial cells. And you'll see from a few of these slides that there's a lot of red, and that doesn't necessarily mean the MSX, which autofluorescent red. Because in the images, there's a lot of background tissue, and these autofluorescent red as well. But it's a darker, murkier red, so you have to be really careful to notice the bright red spots, like you can see here. Those are the MSX. And actually the hardest part of imaging during the confocal microscopy part is looking through all the images and really searching for that bright red spot, because there's just so much noise there. We're looking for the signal, not the noise. So as you can see here, the MSNs, when we finally found the MSNs, were they around the epithelial cells? No. They were mainly around the alveolar macrophages as well, something that we could confirm from the BAALF images from last year. And not only were they in just this slide, but we can see them in all 3 of the time intervals. They're a little harder to see in day 7, but I think now there's a little bit right here. There's some between these cell bodies. I'm going to go out here, and I think there's one on the far right. And then here's a little more clear. You can clearly see the clumps of MSNs right around this cell body, which indicates that obviously the alveolar macrophage is taking up the MSNs. And here, this is a pretty clear one as well. So with this in mind, we did in fact show that over a period of time in the lungs, alveolar macrophages do keep the MSNs. But their alveolar macrophages, the epithelial cells, there's still one more alveolar cell that we did not stain for, the dendritic cell. So dendritic cells and alveolar macrophages, like I explained before, are both immune cells. And they're actually not only similar in just that aspect, they have a bunch of different actions that they share. They're so similar that some may even say that they're the same cell. But with that in mind, our next step of research is to in fact use the protein for dendritic cells, which is cyclic F, and do the exact same procedures that we did for this research and use it for that. Then finally we can know is that if the dendritic cells are the last alveolar cells that may take up MSN. With that, I would like to acknowledge a few people who have helped me along in my research. Thank you to Dr. Pinkerton for giving me this opportunity, helping me out a lot. Big thanks to Morgan, who has basically my rock for the last 6 weeks, always being there for me, teaching me. Big thanks to Dale, who showed me really cool things in the lab. Dr. Reardon and Katie and Dan, who are graduates or undergraduates there, they let us use their fluorescent or their confocal microscope and fluorescent immunohistochemistry materials there. Thanks to Krista, who helped me around, especially the first 2 weeks when I couldn't open the door on my own. And thank you to Dr. Pomeroy and Corinne for this awesome opportunity, and to the whole Pinkerton Lab family, who each and one of you guys have done something different from my experience. And thank you for that. So yeah, thank you. Are there any questions? Yes.
You said you're confocal or focal spectroscopy?
Confocal microscope.
Microscope, it would use the emission of a wavelength. What was your specific wavelength that you saw?
Yeah, so the exact wavelength, man, I'm not sure about that one, but I know spectra averaging 408 is the actual fluorophore, and that whatever wavelength that fluoresces at, that's the wavelength that it was looking at. And actually, obviously, because we had 3 different colors, we also had 3 different wavelengths. So there's wavelengths for each one of those 2. So those are the wavelengths. Yeah. Yeah.
There's something like written on the first slide or something with like PM in relation to MS.
Oh yeah.
What is that?
So that was kind of to emphasize how deep the MSNs really can go. So in the lung research community, we classify particles as, especially in the air, as particulate matter, or PM. So for example, PM10 represents a particle that's 10 micrometers in length. So PM10, as you can see, can only go so far. And the smaller the particle gets, the deeper in the lungs it can get. So, I mean, if you use MSNs in that PM classification, MSNs on average are 70 nanometers in length. So if we convert that into whatever the PM is, the micrometers, MSNs are PM.07, which kind of emphasizes how deep, how small they are, how deep in the lungs they can go. Good question. Yeah?
So if the MSNs can go that deep, is it possible we can go past the alveoli?
Well, actually, the alveoli, they're the smallest parts of the lungs, but actually that's interesting because the MSNs themselves, there's been previous research, like MSNs, we're using them as a drug delivery system, but actually other research has shown that we're using them as a drug delivery system in the lungs, but other research has shown that MSNs actually cross the blood-brain barrier, which is the barrier from which blood can get, or things in the blood can get into the brain. So if we think about it, future applications, this is years in the future, but it's possible that instead of using MSNs for just lung treatments and medicine in the lungs, maybe MSNs can go to the brain, and now we can use them as, drug delivery system for brain tumors, for other brain infections. It's really cool, but there's still years of research to go that far to see how that works. Any other questions? Thank you. Up next we have Krista.
Job more than well done.
Hi everyone, my name is Krista Smith, and this summer I worked in the Pinkerton Lab at the Center for Health and the Environment. We studied the location of mesoporous silica nanoparticles over time in mouse lungs following acute inhalation. Now, mesoporous silica nanoparticles, or MSNs, are an advancement of nanotechnology that are being used as a new and effective method of drug delivery. They can be built for continuous or triggered drug release into specific cells, target cells, without harming normal cells around it. It was found that once inhaled by the lungs, there is little clearance of MSNs 1, 7, and 21 days after inhalation. while we know that certain cells in the lungs are taking up these MSNs, we do not know for sure what the specific type of cell they are. Our goal of our research is to find what the specific type is, because if you look at this DAPI stain here, which stands for cell nuclei, you can see the MSNs around the cells, but DAPI stains cannot tell us what the actual identity of the cells are. So our current hypothesis was that they would not be found in the epithelial cells or the first cells to come in contact with MSNs, and instead we thought that they would pass through the epithelium and be taken up by other cells in the lung, namely macrophages, because they encompass 90 to 95% of the cells in the lung airway. So for this experiment, 17 mice were exposed to positively charged MSNs, 17 were exposed to negatively charged MSNs, and 19 mice were controls and were exposed to no MSNs. Now, once each time mark, of day 1, day 7, or 21 days after exposure was met, a necropsy was performed. During the procedure, the 4 right lobes of the lung were lavaged and removed, and the left lung was inflation fixed with 4% paraformaldehyde and embedded in paraffin. So the embedded left lung was then sectioned into 4 layers, and the 4 layers of the tissue were put onto a single slide. They were put onto a single slide to prepare them for the process of immunohistochemistry, or IHC. Now, IHC provides a visualization of the target cells in the samples by using a fluorescent marker. Now, the way this works is that each cell has a specific antigen that another specific primary antibody will bind to, and then a secondary antibody synthesis of fluorophore of the marker will attach to the primary antibody. Now, this is typically a 2-day procedure, with day 1 focusing on the primary antibody. And in this case, we use CDH1 or ecadherin for the epithelial cell marker. Now, to prepare to put the antibody on, a citrate buffer was used to open up the antigen for antibody binding. And a mouse-on-mouse kit and a Gaussian protein block was used to make sure that the antibody will only bind to the specific target cells that we want it to. Now, the mouse-on-mouse or MOM kit was used because the antibody and tissue that we had were both the same animal. Ideally, the antibody and tissue that you're staining and using the antibody for will be from different animals, but since they were both from mice, this was used as an extra buffer so that the antibody did not find all of the tissue in the sample familiar and attach to all of it. Now, day 2 focuses on the second antibody, and we use streptavidin, and the day 2 also focuses on the DAPI or the cell nuclei staining. And both of these contain a fluorophore, which will create a marker that can be seen under the confocal microscope. So, confocal microscopy works as, in regular microscopy, all layers of the cell will emit light, and in regular microscopy, all of these layers will be seen at the same time, because all of the light, there's nothing for it to specifically pass through. So then these have a blurry and kind of out of focus image because you're looking at so many layers of the tissue at the same time. But with confocal microscopy, you are only looking at one section of one layer of the tissue at one time, because of a plate with a pinhole, which will, since there are different positions of light, it will only allow one layer to pass through. And to move between layers, you would move the pinhole up to then see, like in this image, it would be the top layer if you moved it up, and the back layer if you moved it back. And then the signal on the actual target cells that we want to look for needs to be found with the autofluorescence or background noise of the sample. But typically the signal will be brighter than the background noise, so it is easier to find. And for this experiment, we used 20x and 60°X magnifications because they are both oil-based magnifications, so it is easier to put between them than if we had used an oil-based magnification and a water-based magnification. So this is an image of what you would see in the different channels of the confocal microscopy staining or imaging. And so this is the DAPI or cell nuclei staining. Now, if you look here, and then, well, actually, let me talk about the different things. So DAPI fluoresces blue at wavelength 461, and then the MSNs autofluoresce red at wavelength 596, and as you can see between the two images, they are around these two cell nuclei. And then CDH1 or E-transferrin fluoresces green at wavelength 488. And then typically you would see these in 3 separate channels, and then the 4th channel would composite them all together so you can see where they are in relation to each other. Now, since the confocal microscope can put different layers together, this is the Z-stacking showing the different layers combined that would create a 3D image of that showing the MSN wrapping around the cell nuclei. So as you can see here, the nuclei is showing even though there were only MSNs before, and then there are only MSNs after, which shows that they were completely wrapping around the nucleus. And our main results were that even at the different time points, day 1, day 7, and day 21, there were little to no MSNs found in the epithelial cells. This green staining here was the epithelium, and you can see the MSNs and cell nuclei were not near it, but cell nuclei with the MSNs were not near it at any of the days. And other results that were found were that between the positively and negatively charged MSNs, the negatively charged MSNs fluoresced brighter and were more distinguishable from the background than the positively charged MSNs, because you can see more background, and although here are MSNs here, you can barely tell. You can barely distinguish them from the background. Now, our main conclusion was that the MSNs were not found in the epithelium, and our hypothesis was supported. And so future research would be that we would have to conduct IHC staining on macrophages and dendritic cells, which are the other cells in the lungs, because they were not found in the MSNs were not found in the epithelium. We would also need to measure. We would also need to look at the images that we already had from the confocal microscope and measure the aerial measurements of the cells with MSNs to show the percentages of cells with MSNs and the abundance of MSNs in the cell nucleus. And we would also need to determine if MSN charge really does have an effect on MSN brightness and visibility, and it was not just in those images shown before. So the applications of this is that nanoparticles are in the environment every day in cosmetics and other products, and that there are an advancement of nanomedical technology because they can be used as a targeted method of drug delivery and is very effective at targeting specific cells without harming healthy cells around it. And so I would like to thank Dr. Ken Pinkerton and everyone in the Pinkerton lab for allowing me to work in your lab and have a really good experience there, very informative. And I would like to thank Dr. Colin Reardon and Katie and Dane from the Reardon lab for allowing us to do the IHC there. And I would like to thank Ingrid for her help with the confocal microscope. And I would like to thank Dr. Rick Pomeroy and the YSP staff and students for the opportunity and experience.
Do you know which antibodies you would use for other cells?
So we had originally optimized the IHC staining for all of the cells, the epithelial, macrophage, and dendritic cells. So for dendritic cells, we had optimized with CD11C, and for dendritic cells, we used Cyclic F.
Would the behavior of the MSNs change if they were actually like loaded with drugs, or would it remain pretty much the same?
I'm not entirely sure, but I believe they would be the same because that is why they're used as a method.
Are you making something like here every time?
Yeah, when MSNs are taken into the lung, they just show no sign of leaving the lung, so that would explain it.
Would the MSNs only carry it, or would the lower drugs, or can they do something else?
They can be used for a few things, but we focus mostly on their ability to be able to move through different tracts of the body, specifically the lungs, for drug delivery.